UDP-glucose 4-epimerase, Wikipedia audio. The enzyme UDP-glucose 4-epimerase, also known as UDP-galactose 4-epimerase or GAL, is a homodimeric epimerase found in bacterial, fungal, plant, and mammalian cells. This enzyme performs the final step in the Lewar pathway of galactose metabolism, catalyzing the reversible conversion of UDP-galactose to UDP-glucose. GAL tightly binds nicotinamide adenine dinucleotide, a CO factor required for catalytic activity. Additionally, human and some bacterial GAL isoforms reversibly catalyze the formation of UDP-N-acetylgalactosamine from UDP-N-acetylglucosamine in the presence of NAD+, an initial step in glycoprotein or glycolipid synthesis. Dr. Luis Lewar deduced the role of GAL in galactose metabolism during his tenure at the Instituto de Investigaciones Bioquímicas del Fundación Campoamor, initially terming the enzyme Waldnase. Dr. Lewar was awarded the 1970 Nobel Prize in Chemistry for his discovery of sugar nucleotides and their role in the biosynthesis of carbohydrates. Historical significance. GAL belongs to the short-chain dehydrogenase/reductase superfamily of proteins. This family is characterized by a conserved TRXXX motif necessary for enzymatic activity, one or more Rossmann fold scaffolds, and the ability to bind NAD+. GAL structure has been resolved for a number of species, including E. coli and humans. GAL exists as a homodimer in various species. While subunit size varies from 68 amino acids to 564 amino acids, a majority of GAL subunits cluster near 330 amino acids in length. Each subunit contains two distinct domains. An N-terminal domain contains a 7-stranded parallel beta-pleated sheet flanked by alpha helices. Paired Rossmann folds within this domain allow GAL to tightly bind one NAD and cofactor per subunit. A 6-stranded beta sheet and 5 alpha helices comprise GAL's C-terminal domain. C-terminal residues bind UDP, such that the subunit is responsible for correctly positioning UDP-glucose or UDP-galactose for catalysis. The cleft between GAL's N and C-terminal domains constitutes the enzyme's active site. A conserved TRXXX motif is necessary for GAL catalytic activity. In humans, this motif is represented by TIR 157 GLY Lis-Sir Lis 161, while E. coli GAL contains TIR 149 GLY Lis-Sir Lis 153. The size and shape of GAL's active site varies across species, allowing for variable GAL substrate specificity. Additionally, the conformation of the active site within a species-specific GAL is malleable. For instance, a bulky UDP-GLUCNAC2 N-acetyl group is accommodated within the human GAL active site by the rotation of the ASN 207 carboxamide side chain. GAL inverts the configuration of the 4-hydroxyl group of UDP-galactose through a series of 4 steps. Upon binding UDP-galactose, a conserved tyrosine residue in the active site abstracts a proton from the 4-hydroxyl group. Concomitantly, the 4-hydride is added to the Si face of NAD+, generating NADH and a 4-ketopyranose intermediate. The 4-ketopyranose intermediate rotates 180 degrees about the pyrophosphoryl linkage between the glycosyl oxygen and beta-phosphorus atom, presenting the opposite face of the ketopyranose intermediate to NADH. Hydride transfer from NADH to this opposite face inverts the stereochemistry of the 4 center. The conserved tyrosine residue then donates its proton, regenerating the 4-hydroxyl group. Human and some bacterial Gale isoforms reversibly catalyze the conversion of UDP-glucose to UDP-GalNAc through an identical mechanism, inverting the stereochemical configuration at the sugar's 4-hydroxyl group. No direct catabolic pathways exist for galactose metabolism. Galactose is therefore preferentially converted into glucose 1-phosphate, which may be shunted into glycolysis or the inositol synthesis pathway. Gale functions as one of 4 enzymes in the Lelwar pathway of galactose conversion of glucose 1-phosphate. First, galactose mutaratase converts beta-D-galactose to alpha-D-galactose. Galactokinase then phosphorylates alpha-D-galactose at the 1-hydroxyl group, yielding galactose 1-phosphate. In the third step, galactose 1-phosphate uridyl transferase catalyzes the reversible transfer of a moiety from UDP-glucose to galactose 1-phosphate, generating UDP-galactose and glucose 1-phosphate. In the final Lelwar step, UDP-glucose is regenerated from UDP-galactose by Gale. UDP-glucose cycles back to the third step of the pathway. As such, Gale regenerates a substrate necessary for continued Lelwar pathway cycling. Structure: the glucose 1-phosphate generated in step 3 of the Lelwar pathway may be isomerized to glucose 6-phosphate by phosphoglucomutase. Glucose 6-phosphate readily enters glycolysis, leading to the production of ADP and pyruvate. Furthermore, glucose 6-phosphate may be converted to inositol 1-phosphate by inositol 3-phosphate synthase, generating a precursor needed for inositol biosynthesis. Human and selected bacterial Gale isoforms bind UDP-GalNAc, reversibly catalyzing its conversion to UDP-GalNAc. A family of glycosyl transferases known as UDP-N-acetylgalactosamine polypeptide N-acetylgalactosamine transferases transfers GalNAc from UDP-GalNAc to glycoprotein serine and threonine residues. Pgantase-mediated glycosylation regulates protein sorting, ligand signaling, resistance to proteolytic attack, and represents the first committed step in mucin biosynthesis. Human Gale deficiency or dysfunction results in type 3 galactosemia, which may exist in a mild or more severe form. Tertiary structure. Active site. Mechanism. Conversion of UDP-galactose to UDP-glucose. Conversion of UDP-GalNAc to UDP-GalNAc. Biological function. Galactose metabolism. UDP-GalNAc synthesis. Role in disease.